18 antibiotics. That's how many antibiotics has been developed since 2014. And it's deeply frustrating as a doctor to have 18 antibiotics at your disposal to treat a simple infection, only to find out that infection is resistant to all of these antibiotics. That is antimicrobial resistance. What exactly is antimicrobial resistance? It's a global health crisis that threatens the effectiveness of antibiotics, making once treatable infections potentially lethal. So, why is it important? It's responsible for around 4.9 million deaths annually. There's a diagnostic bottleneck where the traditional methods are time consuming and expensive. There's rising genomic data available, but there's no clinical methods to make it into clinical insights. There's limited availability of novel antibiotics. Since 2014, only 18 novel antibiotics has been developed. So, who exactly is affected? Patients. There's a delay in the diagnosis of the condition of the patient, which leads to ineffective empirical therapy. Microbiologists. There's a lack of timely resistance profiling, so this leads to imprecise antibiotic prescriptions. Pharmaceutical companies. The new drug development by traditional methods is insufficient to meet the demands of clinical treatment. So, what exactly is our solution? GeneHack AMR, an AI based tool that rapidly analyzes bacterial genomic sequences and predicts for antimicrobial resistance. So, how does it work exactly? Well, it's a 4 step process where the first step we input the bacterial DNA sequence. This DNA sequence is from a bacterial isolate. Next, there's a blast system. This blast system rapidly analyzes this bacterial DNA and then correlates it next to a database and gives a list of antibiotic resistant genes. Secondly, it not just the genes, it also predicts a list of proteins that is causing resistance. It gives us the exact mechanism that is causing resistance. Thirdly, it gives us a list of antibiotic list, which is a list of effective antibiotics and a list of ineffective antibiotics based on the particular resistance pattern. This is a demo of our product, GeneHack AMR. So, what exactly is GeneHack AMR? Is that it's a tool that rapidly analyzes bacterial DNA sequence. So, we took an organism called as E. coli with ampicillin resistance. So, ampicillin is an antibiotic. So, this E. coli organism was resistant to an antibiotic called as ampicillin. So, we took the raw DNA sequence, only a short segment of it, and input it into this tool. This AI tool then matched this raw DNA sequence against the database using a system known as BLAST sequence. So using BLAST sequence, we matched it against databases like NCBI and CARD database. Once it did this, it gave us a list of antimicrobial resistant genes. What does this mean? It showed a list of genes in this E. coli organism that was responsible for causing resistance in this specific organism. Resistance to antibiotics. Not just the genes, but it also gives us a list of proteins and their protein sequence. So, what does this tell us? It gives us a list of proteins that is causing resistance in this particular organism, which is depicted pictographically here. Each protein, each gene, along with their protein, which is color coded. Yellow for efflux pumps, green for different hydrolase protein mechanism. So, what does this mean is that these are the different mechanisms by which the bacteria is pushing out or is having resistance to the particular antibiotic. So, this helps a researcher to develop specific antibiotic against these specific mechanisms. So, this is highly beneficial for researchers and also for doctors to understand the exact mechanism of resistance in this particular organism. Lastly, based on the resident pattern that it's analyzed, it gives us a list of antibiotic recommendations. These antibiotic recommendations can be arranged based on the ascending or descending order based on their confidence interval. That means confidence level of a higher number is a much more effective antibiotic. So it gives us a list of effective antibiotics that can be used against this bacteria based on the current resistant pattern. This can also be depicted pictographically using a simple color-coded graph that makes it very effective for a doctor to use and very simple and clinician friendly. Interestingly, it also gives us a list of antibiotics that are ineffective against this particular organism. So now a doctor knows what antibiotic might not work against this particular organism based on its resistance pattern. The real world impact of this tool can be faster diagnostics, precision medicine with targeted antibiotic therapy, and accelerated drug discovery. The future prospects. I understand that this tool may not be highly efficient for a clinician since it analyzes bacterial DNA, but it can be used as an epidemiological resource to track resistant bacterial strain patterns in specific geographical hotspots. It will generate antibiotic recommendations, which can be refined through a physician feedback loop, improving accuracy over time. I believe that over 3 to 5 years, this system can help build a region specific resistance map, which enables more responsible and targeted empirical therapy. Thank you. This is GeneHack AMR.